be you may want to ask. Please, any question that you may want to ask before we start. Okay, so the first part, you look at the cell as a building unit of an organism. And at the end of the lecture, expecting that maybe you should be able to tell us about the cell theory. Then also mention scientists, the names of scientists who contributed to the concept of cell theory. And there are also cell-related concepts like specialization, cell types, cellularity, which we may want to also highlight and wish that you'll be able to explain. So I've given out, there's a proposed out presentation, and we'll look at the levels of organization, the living organism, talk about the cell and the cell theory, then also introduce you to some basic concepts about what the cell is and what it may not. Okay, so to give a background of the cell, the cell is a basic unit of life and actually is the foundation of the structure of an organism. So even with us humans, so when we understand what is happening to the cell, then we'll be able to understand what is happening to the whole human organism, because whatever we experience at any time, especially in sicknesses, it goes back, it goes all the way down to the cell, what is happening to the cell. And therefore, that underpins the fact that cell is a basic structure of an organism. And having known that the cell is a basic structure of an organism, this knowledge serves as a basis for what you call the cell theory. So when you take any living organism, like animals, a whole organism, cells are, we start with the cells, though we have other components of the cell. So on this slide, we have a number of cells. So this, for example, is a nervous, a neuron. This is a neutrophil. We have a skeletal muscle cell. Then we have also a general representative of a cell. Then they come together and they coordinate to form they work together. A number of similar cells come together and they function as what you call a tissue. So here on this slide, we have different types of tissue. Like for example, this can be epithelial tissue, and this can be a connective tissue with some muscles. This is even a bone tissue. Okay, so we have different types of tissues of the body. So a number of tissues of the body also come together to form what we call organs. So we take, for example, the stomach. The stomach is an organ in the human body, and we can find different kinds of tissue. So like we can find an epithelial tissue. We can also find a connective tissue. We can also find some nervous tissue. Then also blood is a specialized connective tissue. Then we can also find some muscle tissue in them. So these different types of tissues come together and they work together to achieve the function of the stomach. Then we have different types of organs in the body coming together so that it can give us what we call organ systems. So we have different organs coming together to give us organ systems. And in animals, especially for especially for mamas. Please pardon me, I have some sore throat, so if I cough, it's not deliberate. So we have different organ systems. So like we have the lymphatic system, we have in humans, we have the respiratory system. We can talk about the digestive system, the urinary system, reproductive system, and so forth. So it's like different organs coming together and they work to achieve the function of the system. So when we talk about the reproductive system, you know that it's for procreation. So a number of organs work together to achieve the procreation. Then you have the urinary system, which will help to produce urine. You have the digestive system, which will also help us to break down complex food substances into simple and absorbable forms. We can also talk about the respiratory system, which will help us to bring oxygen into the body and also take carbon dioxide out of the body. And we can also talk about the lymphatic system, which partly is also involved in fighting, it's part of the immune system. So all these organ systems come together to give us an organism, and you can see different organisms that we see in our environment. So we have the fungi. We can talk about the fish, we have the clams, we have the elephant, we have the tree, which is a plant, birds, we have the starfish, and we also have other plants. But I've been animal biased because we have different, most of the examples that I use are what pertains to the humans. But I think it's also better to use those which are closer to us. But we have examples of plants. Okay. Please, any question?
Say, please, we can't see your screen.
You cannot see my screen.
Oh, we can
Then
see you.
I think you guys maybe from your end.
check from your end.
Exit and join again.
Oh, okay. I've sent it now. Thank you.
Okay. So now we go to the history of cells. So this will introduce us to the cell theory. So we have a number of scientists who played an important role in coming up with this, propounding the cell theory. And one of the pioneer scientists that we can talk about is Robert Hooke. So Robert Hooke is a scientist, and in 1665, by that time we have had a microscope coming in. So one of the breakthroughs in science that push for the cell theory is the discovery of microscope. So by that time, they were interested in looking at structures that are beyond the scope of the eye. So Robert Hooke at that time used a microscope, and he used it to view the thin slice of cork. Cork is the bark of a plant, and with the bark of a plant, it is made up of the dead plant cells. So he was observing the cell walls of plants. Then he noticed that under the microscope, he saw what it's like cells, like small boxes. And these small boxes at that time, it's like the small rooms of that monk's leaf. So, and therefore, he coined these spaces, small boxes, as cells. And so he is attributed for naming cells. So on the slide, I have Robert Hooke, have a microscope that he used at that time, and also what he observed under the microscope. So when it came, people became a little bit curious. You notice that now someone used dead cells, and others became interested in viewing the living cells. And therefore, in from 1665, we now have about 8 years later, another scientist, Anton van Leeuwenhoek. He is also he was a microscope maker, a Dutch microscope maker, and he's accredited to viewing using microscopes to view living organisms, because Hooke used observed the dead cells. So uh, Lee Wan-hook used this simple handheld microscope, and he took pond water and scrapings from his teeth and observed that. So he observed in the pond water, he saw paramecium, and he saw a living organism moving. And you know, a paramecium is a cell, and therefore Lee Wan-hook is credited with the fact that he was the first to view organism. Now, this is an animal cell, and later people also, another scientist, that's Matthias Skleden. Um, Matthias Skleden was also curious that, well, someone has observed their cells, another one is interested in living cells, but he observed an animal cell. And so Matthias Skleden was also interested in what living plant cells look like, not a dead one. So he also observed plants. He looked at plant tissues, and also with a microscope, he was able to identify living cells, those which were quite different from that of Robert Hooke. And therefore, Matthias Skleden concluded that also plants were made up of cells, living, yeah. Then it didn't finish. So we also have a German, Theodor Schwann, who also was so much interested. So he also got animals again, not only Van Leeuwenhoek. So also Theodor Schwann, he also was also interested in viewing animals, and also concluded that animals were made up of cells. So Matthias Skleden and Theodor Schwann, they ended up saying that plants are made up of cells, animals are also made up of cells, and they contributed also to founding the cell theory. Now, as years progress, it became a question that so where does cells originate? And this is where a scientist, Rudolf Virchow, the works of Rudolf Virchow became very, very important. So he was a medical doctor, a German medical doctor, and he used a microscope and was observing. He regularly observed the cells under the microscope. Then he realized that cells, he saw under the microscope cells which were dividing, and when he saw these dividing cells, then he came up with he reported that actually cells come from pre-existing cells. So you have a cell, and then later the cell is going to divide and give rise to another cell type. And all these activities of these scientists, the reports of these scientists, are what form the basis of cell theory. And in the cell theory, we can talk about three main facts. The first is that all living things are made up of cells. And two, the cells are the basic unit of structure and function in an organism. Or you can say that the cells are the basic unit of life. And the third one is that cells come from the reproduction of existing cells, or you can say that cells come out from pre-existing cells through cell division. So these 3 points is what make up the cell theory. Okay. Well, after the cell theory has been propounded, later as people became more interested in cells, and we noticed that cells are made up of organelles. And in about 1970, we have a theory which came out, symbiotic theory.
Hello, sir.
Yes.
Hello, sir. Sir, good morning.
Yes, I'm listening.
Sir, please, I know
Morning,
that
sir.
I'm not supposed to distract you, but forgive me and help me fill my
Oh,
curiosity. Yesterday we had lectures on cytology and genetics, and it seems what
okay.
we are doing right now was the same thing. May I know cytology and then cells and tissue? their starting point, are they the same, please?
Oh, okay, so you didn't join the early part of the
Oh,
lecture.
okay,
So, like as an
okay,
introduction,
okay,
so I mentioned
okay,
that this course will focus more on the tissues. We'll focus
okay,
more on the tissue
okay,
because basic cytology will also cover this. So what we do
okay,
is that, but what we do to complement the cell is that as we're going through the tissues, we'll mention some cells of the body.
okay,
But as to
okay,
the details of the cell, the organelles, the organelles like what the mitochondrion and the other things are behaving, for that we wouldn't give the details.
okay, okay. Thank you, sir.
So at this point, they all be similar, but beyond that, that is it.
Okay,
I
okay.
hope
Thank you.
you are okay.
Yeah, I'm okay. I'm okay.
Okay, sir. Okay. So we have the endosymbiotic theory, where American biologist called the Lynn McGuinness. So he also she worked and she was also doing some research, and she also reported that actually some organelles in the cells were some time ago were free living cells, then they became incorporated in other cells. So hence they were endocytosed into other cells. So endo is internalized, and as they were, can we get please mute your mic? Okay. So these cells were once living, they were individual cells living outside, and later they were internalized into the cell. So much more like they were bacterium, especially the bacterium. We have the aerobic bacterium and the cyanobacterium. So these were internalized into various cells. So after internalization, we have what you call the mitochondrion, then in which is present in both animals and plants. Then we have the also chloroplasts, but they are found in primary producers, the cells of primary producers. So the endosymbiotic theory also came into light, and these were happening because they noticed that it's also possible for the mitochondria and also the chloroplasts to have their own DNA, which is quite different from the cell. So this tells us about what happened to bring about the cell theory, and also beyond after the cell theory has been propounded, a theory that came after, that's the endosymbiotic theory. So I will move on to tell you about some basic concepts about cells, and one of them is the size of a cell. So on the slide is displayed, we have various living organisms. We have organisms like various structures, including organisms, especially organisms, and also cell. And all these, we have a range within which the human eye can see. And we also have some, like, for example, from 1 millimeter even as big as like an organism like the whale, 1 kilometer, it's very easy for the humans to see with our eyes. Then we have below 1 millimeter, at least we can also still see. Then we have the human egg. You can see the human egg. You need, beyond this range, you need the microscope. So the light microscope comes in to help us to see up to below 1 micrometer. And even with the light microscope is cannot also help us see structures that are in the nanometers, like 100 nanometer up to the 0.1 nanometer. So what it means is that the the components of the cell which the organelles within the cell, which of which information, the information that we have about the components of the cell, we got most of them using the electron microscope. We got the structure mostly through the electron microscope. Now, what is the cell of the size of a cell? So we can talk about cells ranging from 5 to 50 microns. And a number of, if maybe if I have to talk to you about microns, microns is like you have 1 centimeter on the ruler, which we are familiar, and that 1 centimeter, you divide them into 10,000 parts. You divide them into 10,000 parts. No, and also, if you are familiar with inch, 1 inch is about 25,000 microns. So you have 10,000 microns, like here, 1 centimeter. You divide it into 10,000. Then you just take about five. You take five. So you notice that it's even lesser than 1 millimeter, and our eyes can see about 1 millimeter, and less than 1 millimeter, it becomes a problem. And that is the nature of the cell. That's the cell size, 5 to 50 microns. Then when it comes to organisms and their cellular composition, we bring I'll tell you about cellularity. So we have what you call unicellular organisms and multicellular organisms. So in unicellular organism, the word uni refers to one. So this refers to organisms that is just made up of one cell. So we have organisms like the protists, like the amoeba, we have the paramecium, we have also other organisms which are made up of multiple cells. So we have also multicellular organisms, and they are made up of different kinds of cells. But one thing is that with multicellular organisms, the cells usually assumes different roles. And when they assume different roles, we use the word specialize. So cells assume different roles. And one of the things that even evidence that suggests that cells have taken different roles is the shape. So you look at the shape and you see that cells, there are different shapes, and it tells us about at least that these cells will have different functions. But in multicellular organisms, the key thing is that there are different types of cells, but they are all not performing one function. They perform different function, and they are able to do so because they undergo what you call differentiation or specialization. So in cell specialization, what happens is that, a cell, a particular cell, every cell in the human body is made up of the same DNA. It's made up of the same DNA, but the different cells will have to activate certain aspects of the DNA, which we say that that's the genes. A segment of the DNA will comprise what you call the genes, and different cells will activate the different genes within different times. So on the slide, this is the picture of a normal a cell which is giving rise to different cells. Yeah, so the DNA in the or the genome, genetic constitution of this cell is the same as all, but just that in these cells, they are different genes are turned on so that we can have this kind of cell. In this other cell, different genes are turned on so that we can have another this kind of cell. Then also the same applies to the third one, and even the cell which is giving rise to the others. And in science, we take advantage of this so that nowadays we try to do some kind of staining, and we pay attention to the cells, the proteins, the genes which are expressed. And I'm sure when you do the cytology and genetics, it will come into mind transcription, translation, and protein synthesis. So we take advantage of the specific proteins that these cells are expressing in order to identify the different cells. Okay, so in the animal cells, We have later I'll introduce you to this kind of cell, which is a muscle cell. This is a skeletal muscle cell. This cell has nucleus, it has nuclei, it also has cytoplasm. Then we also have the red blood cell, which looks like this under the microscope. Even we can also get the scrapings of our cheek, the cells, and if we apply stain color to it, this is what they look like. So you notice that these cells are having different shapes, and the underlying fact is that there's different genes are activated at any particular point in time to be able to derive these cells. When it comes to plant cells, we have what we call the guard cells, and these are present in the leaves. They will regulate how much light and light will enter the cell, or the oxygen will move into the cell. We also have the xylem cells for transport of water, and we can even talk about the phloem cells, which are also specialized in transporting um, food substances. Okay. Then we have the what is also very important for um, procreation among plants. That's a pollen grain. Yeah. So the pollen grain is, is male. So this is also another type of cell. But all these cells, the underlying thing is that they are coming on as a result of specialization. Then I will also talk about the different cell types, the different cell types. So in our body, we have the different cells. So, and these cells are similar to the eukaryotes, like the animal, the kingdom. So we have the prokaryotes, and we also have the eukaryotes. The prokaryotes, we can talk about the bacteria and the archaebacteria. Then we have the eukaryotes, we can talk about plants, we can talk about fungi, animals, and the protists. Usually the protists tend to, yeah, so these are all eukaryotes. Okay. So we can talk about the plant cell, fungi cell, the protist cell, animal cells, the bacteria cell, the archaebacteria cell. So, but nonetheless, we also have what you call prokaryotic cell and the eukaryotic cell. So the different cell types which are present in the world stems from the kingdom classification of organisms. So I'll briefly talk about the eukaryotic cell and the prokaryotic cell. So in the eukaryotic cell, cells have a nucleus, a well-defined nucleus, and also a membrane, and the nucleus is bound by a membrane. Then we have also other organelles which are having a membrane that binds them, that bounds them. So they have membrane bound organelles. So, and they have more complex, this more complex type of cells. Then we can also have the prokaryotes. Prokaryotes don't have a well-defined nucleus. So usually the nuclear material is scattered in the cytoplasm, and that distinguishes the eukaryote from the prokaryotes. So this will bring us to the end of that's an introduction to the cell. Okay. So maybe I will change the slide, and any question, I will change the slide, and we'll now talk about the tissues. Hmm. Please, any question?
So I think there's a hand up.
Okay, yeah, please, you can go on. Please, you can ask a question.
Please, a minute. Hello, sir.
Yes.
Please, can you go over the function of the guard cell for me, please?
The guard cell.
Yes, please.
So I said that the guard cell is a specialized plant cell which allow gases. It regulates the gases that are entering the cell, the plant. So there is a plant tissue, and within the tissue, all gases should be able to move through the tissues. Okay, so the guard cell is specialized in regulating how much oxygen and carbon dioxide will enter the tissue or also leave the tissue.
Thank you, sir.
Okay. So maybe briefly we would look at okay, just a moment. I think I'm struggling to upload the slides. What's happening?
So what is happening?
The lecture
I think dog is
is off.
awful.
Hey, Asema.
Please, can you confirm whether you can hear me?
Yes, sir. Sir, we can hear you.
Okay.
Yes, sir.
Yes, sir.
Okay,
We can hear you.
Yes, please.
okay. Sorry about that. Okay. So um with the second lecture, we'll talk about histological techniques. Histological techniques. So, histology. Before I make that comment, with this lecture, we are hoping that you will be able to understand the steps, explain the steps which are involved in what you call tissue processing and also tissue staining. And I will introduce you to the histological methods, the tissue processing, the tissue staining, and also types of specimen preparations. So, we are at the tissue level. For us to know the details of the tissues, we need to process the tissue and also view them under the microscope. So this brings us to a term where there's in the discipline of science that talks about that studies tissue is what you call histology. And one thing is that usually the tissues which is made up of cells and also the constituent of cells, when you observe them under the microscope, most of them are difficult to see. They are colorless and they are virtually transparent. So, and light passes through easily. It makes it very, very difficult to also distinguish among the components of the cells and also components of the tissues. And therefore, it became necessary that, but one thing is that when we apply dyes to them, then this kind of limitation is overcome. So, when we notice that, then it's led to scientists, especially like histologists, developing a wide variety of different staining techniques, which will identify different elements within the tissues. So when you go to the lab, any histology lab, you'll be seeing this kind of things. This is a slide box, and you find this is a glass slide. On it, we have a tissue which have been processed and stained. We put on it, then we put a cover slip on it. So this is like a final product, which we place under the microscope to be able to see. You cannot just put maybe a slice of onion under the microscope. If we have to be mindful of the thickness that we can make out of the slices and view them under the microscope. So this is the lecture. In the lecture, I will let you know how we come about these glass slides, which we use in studying the component of tissues. So I will let you. There are some terminologies that I think you should be familiar with. So we have histology when you are studying the tissues and their structure. That's the normal tissues. Then we have the histopathology when you look at the tissues in their disease state. How are they appearing? So that aspect of science looks at the histopathology. And we also have a term histotechnique, which is the techniques, the processes that are involved in processing the tissue and also like staining them. Okay, so the techniques for processing the tissue, whether biopsies are some pieces of organs which are obtained from a living organism. So and when in humans, we can take them from surgery. Sometimes people in autopsy, we can take small pieces of this tissue and go and process it so that we now study them under the microscope. Now, the basic next is that the basic histological methods, we can group them into two phases. We have what you call the tissue processing and tissue staining. So now what happens, the tissue staining. So you look at the tissue processing, but this comes from the principle that what happens in tissue staining. Now, when cells are living, rarely would they pick up stains. Rarely would they pick up stains. So we need to process them into a state where they can easily pick up stains. Okay. Now, the first step is the tissue processing. Now, the aim of this first phase of the histological methods is to put the tissues that we want to view under the microscope in a background medium, which will give the tissue some form of rigidity. The human tissue and also plant tissues are very, very soft. For plant tissue, because of cell wall, so sometimes you can have it, you have the wall a little bit strengthened. They have a firmer structure compared to animal tissue. Animal tissues are relatively fragile. Okay, but in the first phase of the histological methods, the whole idea is to put the tissue in a background medium, which will give some form of rigidity to the tissue, and it makes them, and that rigidity is also such that it allows us to be able to cut very, very thin sections of thin sections of the tissue and put them on a slide. And these tissues are also not hard, very hard or rigid to damage the knife that we are using to cut the tissue. And we can, whenever we take animal tissue or plant tissues, we can process the tissue either fresh or in a fixed state. In a fixed state, usually I will introduce the paraffin concept of paraffin. So I'll mention that there are steps involved in processing a fixed tissue. So we have what you call the fixation, we have dehydration, we have clearing, we have infiltration or impregnation, we have embedding, then we have sectioning. So this is what I'm going to talk about. I'll tell you the details of each steps, and we can also and to aid in following, I've placed a snapshot of these processes on the upper right corner of the slide. So first is to talk about fixation. So one thing is that whenever we have any tissue and we place them on, we remove them from the living organism and we put them down, what happens is that with time they undergo putrefaction, which is decaying. And in the case of for plants, the smell is not so bad, but animals, they can give out a very pungent smell. And in these, if you want to observe them under the microscope and immediately we allow the putrefaction to happen, we lose it, distorts the original structure of the tissue. So usually you want to observe a tissue under the microscope, you don't allow it to undergo decay, because if you allow it to undergo decay, the structure will be gone. So it is important that we place them, we fix them, so that at least we can maintain the tissue in a lifelike state. Now, what brings about the putrefaction? This will introduce me, I want to introduce you to a term called the autolysis. Autolysis. So autolysis alters the structure of the tissue. What autolysis is, it comes from the word auto and lysis, which is a self-decay. Now, usually when a cell dies or you take it out of an organ from the living organism where its life is sustained, within each cells, there are organelles like the lysosomes. We have organelles including the lysosomes, and they release enzymes. These enzymes, whenever they are released, they will leak into the cytoplasm. And when they are in the cytoplasm, these enzymes will digest everything of the cell. They digest the components of the cell, and that is, it hydrolyzes the components of the cell in a process called autolysis. So this is the underlying principle for putrefaction. So when we do fixation, the purpose of the fixation is to inhibit autolysis. The purpose of fixation is to inhibit autolysis. And in that case, we are able to maintain the tissue in a lifelike state as possible. Then also the fixation is also there to also prevent dehydration. It prevents the tissue dehydration because when you allow organs to lie down, they lose water with time, and with time they become very, very dry. Then the fixation also hardens the tissue so that at least it is easier to cut. And the fixation also protects tissues from pathogens. And finally, it also allows the tissue to be able to retain dyes, because later we want to apply some stains to them. So it helps them to retain the dye. There are methods of fixing tissues, and we have chemical and physical means. So physical methods, we have heating, microwave, and sometimes we put organs in fridges. And these are methods which we are very familiar with. I'm sure that in our homes, when we buy meat from the butcher, if we don't have a freeze, a refrigerator to freeze, a freezer to actually freeze this meat, you notice that one of the common things that they do is to actually steam the meat, because we don't want it to go bad. So when steaming and applying heat is one of the methods for fixing tissues. But usually with our biological studies, we rarely use heat fixation. The commonest one is the chemical means, and we have some solutions that are able to perform the fixation. So for us to be able to do fixation, usually when we are processing the tissue, like you have liver, you cannot take the entire liver, you only cut a piece of the liver. And in the lab, we have what you call the cassettes. We put the liver in this cassette, then we close it so that we get this particular state. This we call the tissue cassettes. Now, even when we are placing them in the solution, in this cassette, it helps us to be able to lift the organ slice out of the solution easily. Unlike if you want to put the organ in the solution, whilst you are using the forceps, the tool to bring it out, you can also damage the tissue. Now, in the lab, there are a number of fixatives which are available, but the commonest of the fixative is formalin, is formaldehyde, and we prepare a 10% concentration of this formaldehyde, and we use it for fixing. That is the popular fixative that you can ever think about that we have. Other than that, we also have like ethanol or methanol, 70% ethanol or methanol. We can also use it for fixation. Sometimes we use acetic acid for fixation. The methanol is actually the alcohol is mild form of a fixative. So usually in a cell, when we are in science, we can also culture some cells and put cells on glass slide and fix them. You can put methanol and it can fix the cell. Then we also have acetic alcohol, which is also a very good fixative. Now, beyond this, what happens that sometimes we can also combine these fixatives with other substances to produce a fluid. So we have what we call Zenker's fluid, Hellis fluid, Bowen's fluid, Canois, and cold acetone. But the commonest of it is the Bowen's fluid. So, and the dehydration, the fixation can take about, let's say, 24 to 48 hours. Usually, if you are processing animal tissue, we put it in about the solution about 48 hours, a minimum of 48 hours, because we also are so that it will allow sufficient time to get fixed. Then we have the dehydration. Now, dehydration becomes necessary because we want to put the organ in a solid medium, but a solid medium is not miscible with water. And the part of the fixation, the importance of fixation, one of the benefits of fixation is to hydrate the tissue. And before we can get to a state where we put the organ in the embedding medium, what we do is that we have to take out the water. So the dehydration is the removal of water from the processed tissue, from the fixed tissue, and the purpose of it is to allow complete penetration of the tissues with the embedding material. We have to bear in mind that so long as there is one thing is that so long as there is water in the tissue, it is subjected to bacteria breakdown or decay, and therefore the dehydration also prevents that. Then we also have when we dehydrate, it makes the tissues a little it appear opaque, and so that can give it at least it can limit the amount of light that goes through. In the lab, the commonest way of common method for dehydration is to use alcohol, ethanol, especially. Other alcohols are weak. Ethanol is the best. So we have various containers, and in those containers, we prepare for one container. We put 10% ethanol in it, another 20%, then 70. We have 95 or 90 percent ethanol. Then we get another jar where it is only 100% ethanol, 100% ethanol. So with the tissues in the process tissue in the cassette, we will place them in succession. So from 10%, after some minutes, we put them in 20%. After some time, we put them in 70%, then 95%, 100%, and 100%. Now the whole idea is to not we don't put it straight into 100%, because when you put it directly into 100%, the dehydration process can disfigure the tissue. So we do the dehydration in a stepwise manner. Now, in this step, sufficient time is usually allowed so that it will allow for complete extraction of water. And the whole, if dehydration is carried out too rapidly, it will bring about distortion and sometimes the shrinkage of the tissue. After dehydration, the next step is clearing. The next step is clearing. Now, even with the embedded
Hello, Doc.
medium,
Hello, Doc.
yes,
Please,
yes.
with the dehydration using ethanol, you made
Yes.
mention that you have to put them in assessing form, like from 10. Is it the same tissue that you are using that you pass it through the 10, 20, 70, finally 200, or variety, different tissues?
Oh, okay. So this is the tissue that is being processed.
The same
So,
tissue that is being processed, you put it in 10, remove,
and after
put
some
it
minutes, you
in
transfer it
20.
into 20, you transfer it into 70, 95, 100, 100.
Okay, okay.
And usually this protocol holds for all tissues. So you can, so the tissues are in the cassette. Sometimes we don't necessarily even put one. We can process different organs, different tissues of an organ tissues, but they are small. We cut pieces of them and put them in one cassette. I hope, I don't know whether you get a picture.
Yes, please. Thank you.
Okay. We can process tissues together, but the only time you separate them is when you are embedding. Sometimes people embed them individually, even that you can even put them together, because under the microscope, we are able to distinguish the various tissues from one another. Okay, so then we have clearing. So in clearing, you see, our ultimate goal is to put the medium, the tissue in a solid medium which will give it that rigidity. And even that medium, the medium is not soluble in the alcohol, the ethanol that we use for dehydration. And therefore, what we do is that we try to replace, we leverage on the fact that there are some solutions that are soluble in the ethanol and also soluble with the embedded medium. So we bring them as intermediate solutions so that we can use it to replace the ethanol. And when we are taking out the ethanol from the tissue, then that brings us a process. That is what we call clearing. And also the clearing is also such that when we finish this step, the tissue becomes more transparent. Before after the dehydration, the tissue is opaque. But when we take out the ethanol out of it, the tissue becomes even transparent. And that is where even the process of clearing came about. So I've indicated the point. So this step is necessary because the dehydrating agent, which is the ethanol, is not miscible with the embedding medium, which is a medium that is supposed to give rigidity to the tissue. And therefore, we introduce an intermediate fluid that is miscible with both the ethanol and also the embedding medium. Okay, so there are a number of clearing agents, but in the lab, histology lab, the commonest that we use is chloroform. Chloroform is a popular chemical for comatosing organisms, making people comatose. But also, chloroform has, we also use it for the clearing step in tissue processing. There are also other clearing agents. Sometimes we hear of xylene, we have toluene, we have clove oil, we have benzo, cedarwood oil, depending on the protocol. A particular protocol for what we need a protocol for. They would prescribe a particular clearing agent to use. Nonetheless, the commonest is the chloroform. So after clearing, the next step is infiltration or impregnation. And we want to put the organ after clearing, we want to put the organ in a solid medium. However, because for the sake of doing it gradually to get a perfect rigidity, we pre-treat the organ, the processed organ or tissue with the embedding medium. And this step is important because one, the embedding this step infiltration will get rid of the clearing agent, and two, this when the clearing agent is removed, the embedding medium will infiltrate, will fill all those holes, openings within the tissue, so that at least the rigidity will not just come from external, it is also coming from internal. So in the lab, there are a number of embedding medium. So in the microbiology lab, they use, for example, agar. Agar is one medium which provides, it is a solidifying medium. Then we have also gelatin. Then we have wax. Okay, so when this medium, they can, in a particular temperature, they can be, they are mostly liquid. Then in a lower temperature, they are warm, they become solid. And that is where they provide external support. They make the organ more hard. Now in the lab, the commonest medium that we use is a paraffin wax. Is a paraffin wax. And the paraffin wax is actually what makes up what we use for candle. So this is what the same thing that is used for candle. And this is a mixture of solid hydrocarbons which are being produced during the refining of coal and mineral oil. Usually the wax is solid in temperatures below 39. In temperatures below 39 is solid. And within 39 to 68, the wax melts. The wax melts and it is in the liquid state. It is in the liquid state. Then beyond 68,
Doc, please, can you go over the agar for me, please?
the
Beg.
agar
Yes, please.
is a solidifying, it's a medium in the lab, and it is used to solidify, to harden structure. When agar is cold, at a lower temperature, agar is hard, it's a solid. But in a higher temperature, the agar is liquid. Okay, so if you want to embed the organ, what you do is that you first make the agar liquid, then you put the organ in it, you allow for sufficient time, then you now allow the agar to cool. So by the time the temperatures fall below the melting point, okay, it's a solid to liquid, so we talk about the melting point. So by the time you at that lower temperature, then the agar will solidify, and that will give the organ that rigidity. Please, is it a little okay?
Yes, please, Doc.
You
Thank
see, in the lab, when we are doing when you are doing all these things, wax is agar is more expensive compared to wax. Wax is relatively cheaper. So if we have a method which is cheaper and can give us the same results, we go for the cheaper ones. So in this, that's why one of the reasons why wax is used often in embedding. Is there another question?
The gelatin too, if you can explain it in
Oh,
English.
gelatin is also one of the solutions. They all work in the same principle. In a particular lower temperature, they are solid. In a particular temperature, they are liquid. So when you want to just use them, usually the principle is to just raise the temperature and make them liquid. Then later you cool them, then they become solid. So it's the same thing. So in the liquid state, you put the organ or the tissue being processed in it. You allow for sufficient time, then then you cool it, and when you cool it, the gelatin becomes solid again, and it will give the organ that firm structure. So it's the same principle. Okay, so but before you do the embedding, you have to do the infiltration step with this embedding medium. So wax is this. It has a melting point of 39 to 68. So when you have 39 to 68, the wax will be in the liquid state. So in the lab, I think we usually use about 50 degrees. Now about 68, the wax will be burning. It gives a choking smell, and that choking smell is not very good for our health. So within these particular temperatures, not that you cannot keep it in 100, but even when you keep it in 100, it means that you are burning the wax, and the smell that comes out is very, very pungent, and it's not helpful. It doesn't support human health. So hence we stick to about 39 to 68, and averagely most of them will use about 50 degrees oven. So they set the oven in 50 degrees, and when they put the wax in it, the wax will melt. So the wax as an embedding material, we have a number other factors that we use that makes us to use the wax as an embedding material. So we've also we add other substances to it so that it will even improve its utility as an embedding material. Now, when one thing is that when the organs are put in wax and they are given a solid support, later we'll be cutting. And when you cut tissues, one thing is that it helps. We have what you call rebonding, where various cuts will be continuous with one another, will be joined end to end with other cuts that we will make. And paraffin is able to give us that kind of result. So it's and they hold they are held in contact with one another, and that is the benefit of the wax, using wax as the embedding material. Okay, so when you are doing this step, we also watch out that there are no dust particles in it. So I'll take you through how the bending is done in the lab. So we have another use for the cassette. So we use a cassette to hold the tissue which is being embedded. So in the lab, we have a mold. These are made up of aluminum. Those mold, and we have a space where we can put the organ that we want to embed, and we pour the wax around, molten wax. The wax in its molten form, we pour it around it. Then we place the cassette on it, and we pour more wax on it. So by the time we put it on ice, and by the time the organ is cold, the setup is cold, we will have the wax stuck onto the this platform, of which is gotten from the tissue cassette. So you can just take the bigger portion of the cassette and use it for this purpose. Coincidentally, this cassette fits into the cutting machine, and hence it encourages us to use this. Now, when we are embedding, we are also mindful of the kind of cuts that we want to get. So sometimes we have an organ which is tubular, and we want to get the TS section. So when we get that, we have a way of placing it so that after embedding and removing the block, when we cut, we can still get the TS section. If it's an LS section that we want, we have a way of placing it, the tissue in the mold, so that after the embedding, when we are cutting, we will get the TS section of that. So this is end product of when we do embedding. This is what the kind of things that we get. You see some white outline. Those are parts of the cassette. The cassettes come in different colors. Some have it in pink, others have in blue, others have in white. So the colors are varying. So like you want to get a section like this, we have a way of placing the organ in those mode. So this is a magnified picture of the various blocks of tissues that we get after embedding. And they can stay there for a long time. So long as the temperature is cold, it doesn't melt it. You can have this wax for a very long time. The next step is sectioning. This is a step that we will be cutting. So this is a step that we will be cutting. So we've had a firm, an organ. The organ is in a firm state. We need to make pieces of cuts, and usually we cut about 1 to 50 microns with a knife. The knife is a blade, but with this paraffin embedded wax tissue, we have a machine for cutting. We call it the micro tool. So there's a picture of the micro tool. It's like a sewing machine. It has a part that you can put blade, and we have also a slot for holding the tissue. So we hold the tissue here, and we put the blade there. Now, as it also operates on this revolving, as the handle goes through one revolution, then the blade will also make some movements. It goes towards some distance, so that as you make several turns, then the blade, the part holding the blade will be moving in that distance. Then you'll be making your cuts. We also have other machines for the section, and we have the vibratome, and cryostat. But usually the vibrato, this is the vibrato, and it is usually used to cut when we need sections up to 200 microns. So if you want to cut sections which is more than 50 and it's about 200 microns, advisably we use a vibrato. It also works on the same principle, just that it is more robust to cut thicker sections like 200 microns. It's very, very thick. Though it's small in our eyes, but for the tissue, it's very, very thick. And it can be used to cut fresh or fixed samples. But in the case of cryostat is for frozen on fixed tissues. So we have a tissue that is frozen. And if you want to make very, very small cuts, like what is used for the microtome, we use a cryostat. This is a picture of the cryostat. So the cryostat is like a freezing compartment. This is a freezer. But and you can see the rotary part. All of them have the rotary part. Whenever you are moving, you move it through some revolutions. Then the blade will also be moving through some distance. When you look at the inner aspect, this is the part that we use to hold the tissue. And then we have a blade where we fix the blade. Okay, so we rotate it. And as we rotate it, the blade will be moving through some distance. Actually, in Ghana, I think maybe this is an expensive gadget, very difficult to get. I don't know how many where you can get a cryostat. Okay. Now, so I'll go back to section of the paraffin embedded tissue, and in the paraffin embedded tissue, when we are cutting, we put it in a block and we are cutting. So this is where we place the tissue, then the plate. So once we are revolving, it will be cutting, and we get a ribbon of these tissues. We get ribbons of these tissues. Now, next step is that we usually put them on a water bath because the paraffin wax, there can be fold in the tissue. There can be fold in the tissue, so we put them on the paraffin a water bath about 37 degrees Celsius, not too hot. If it is too hot, you put the wax, the ribbon on it, and it is going to melt. So about 37 degrees or maybe a little lower, the wax would spread out and it will prevent folding within the tissue. Then it also allows us to be able to break the ribbons into pieces. So you break the ribbons into pieces. The next thing is to use a glass slide to fetch the ribbons one by one. Okay, depending on the size of the ribbons, sometimes it becomes necessary to put maybe two. You can pick two on it, but usually we put one. Then we allow it to dry, and then it is now ready for the second section, which we call the staining. Please, any question?
So this is really practical, something that we are also doing. And I think if there will be some aspect where we can see it, this is really technical because I'm trying to follow the steps and then trying to understand. But it is new when it comes to some of the techniques that you have to apply. So I don't know whether something can be done in terms of practical something so that we can also
For now, it will be kind of difficult to make that arrangement. Yeah, we have to do some checks, and so, but we'll do our best to get you some videos, which currently, if I had it, then I would make. If I had this, at least a microtome, I could have made the arrangement for you to do that. But the microtome is not there, so it's the others you can put it in you can make a provision. Yeah, but for the microtome, that's a machine that it's not easy to come by. I don't have it in my lab, in my department for now, so I have to outsource it. But what you do is that maybe it is possible to fuse the tissue processing into another practical that is being done. That if we are going to do a practical, we can fuse it into it. Yeah, so maybe we can do that, but maybe not in this module, not in this course.
Okay, sir. Okay, thank you.
Okay. Then we have the staining. So with the staining, this is
Say,
yes.
hello, sir. And so, per what my brother said, never say na eja. My boy and kakra in the exams, no send we will not find ourselves wanting. I mean, the question, how the question will be. Now, a bit it's a class that we can also maneuver through. A
Some people may be interested in what you are saying, you know.
But
right.
Festus, you again. Festus, you again.
Yes, sir. Can I ask a question, please? Please, I would like to find out if this lecture is there's a background noise we can't hear.
Okay. So okay, thank you. So we have the next step, which is tissue staining, and this is when we apply color to the processed tissue, so that we can view them observe them easily under the microscope. So in staining, we apply artificial like colors to substances so that it will allow for examination of tissues, microorganisms, or even other cells under under the microscope. So it comes back to the point that tissues are transparent. The cellular elements, they are transparent. So um, when you don't apply the dye, you place them under the microscope, you don't see anything. You cannot distinguish among the components of the tissue. So hence the step. Now, cells, they are in the lab, there are common types of stains. There are different types of stains, but what we routinely use is a hematoxylin and eosin. Hematoxylin and eosin. So in this stain, the stain is more of a chemical reaction. The stain is more of a chemical reaction, and in this chemical reaction, it's mostly also based on acid and bases. So we have hematoxylin and eosin. Hematoxylin is a base, and it binds to acidic structures and it stains them blue. In the cell, the structure that is likely that contains a lot of acids is a nucleus because of nucleic acid. So if we introduce DNA, RNA, they are all acid in nature. And therefore, when we apply the hematoxylin binds to the acid and gives the acid the bluish color. Okay, so we use that to give the nucleus that color. So because the nucleus is acidic and it loves picking bases, we usually say the nucleus, we usually refer to it as a basophilic structure. Then the other one is the eosin. Eosin is acidic in nature. So, and it stains basic structures red. And in the cell, the cytoplasm is slightly basic, and therefore it picks the acidic color. And therefore, we see that the cytoplasm is acidophilic because it's base and it likes picking acids. So these have been the principles so that we are able to, when we are picking some stains, we pick a stain that can react to the nucleus. Then we pick another stain that can react to the cytoplasm. And when we look at it under the microscope, the outline of the cell easily comes out. So this is
Okay. So it
hello.
means we can't add them at the same time.
Yeah, so it is stepwise.
Oh, okay.
It's done stepwise. So this is a picture that of a tissue that was processed and stained using the hematoxylin and eosin. This is how the kidney appears under the microscope. So you see that there are some regions which are having this blue or purple coloration, the circles. Those small circles are the nucleus. So the nucleus, and even with the nucleus, there is a nucleolus. So you see that for some of them, the dots, there is a dot in the center. Those are the nucleolus. So when we do that staining, we have that as a nucleolus. Then in eosin stains the cytoplasm pink. Eosin stains the cytoplasm pink. So you see that around these structures, you have the pinkish coloration. You have the pinkish coloration. So this is a cell. This is also another cell. So this is so you look at the nucleus and the surrounding region, and it's able to let you know that this is a cell. This is also a cell. That's also a cell. Now, we have to bear in mind that during the cutting, the cells are actually 3D. They are 3D. And when you are having a 3D, depending on the part that the blade will go through the tissue, those are the structures that we can see. So in some of the cells, you can see the nucleus, but areas like this, you may not see the nucleus. There are things, a possibility. It's possible if you are looking at the disease state, it may be possible that those cells are dead, so you don't see the nucleus. Or it can also be that the blade couldn't go through the nucleus for us to be able to see that part. Then we have other cells which are very, very flat. So you see the nucleus, but their cytoplasm is very, very flat. So usually you see it will look as if that the nucleus is also just there. Okay. Then we have other situations where the cytoplasm, there are also other things that are secreted from cell, which the EOC will pick. So, for example, in this tissue, you have a lot of fibers. There are fibers that are being released, and the eosin pick up the eosin, and therefore you may not see the complete outline of cells, unlike what I showed you in this region. Then there are also other special stains. There are also other stains, but usually they are relatively more cost involving, and they are for specific purposes. And we have stains like periodic acid shift, Mason's trichome, Golgi stain, nasal stain, lipofuscin stain, and methylene blue stain. And these are stains which are meant for a particular purpose. So depending on what we want to study in the tissue, then we will use these stains and we can compare among groups so that we can understand what the whole process, what is happening in the tissue. Now, all these things that I mentioned are they are a typical example of histochemical staining. So it's like a reaction between a chemical in solution and also the tissue. But however, recently we have also another staining technique has come up. Not to it's quite old, um, but this staining technique is based on an antigen antibody reaction. So if we want to detect a particular protein in a tissue, we develop antibodies against that particular um, antigen. And in the course of the staining process, we will introduce the antibody to the antigen, then they will react. Now, attached to the antibody is a color expressing substance, so that for those substances, when you expose them to light, they give off a particular color. And because the antigens, and also the antigens are restricted to specific segments of the cell or tissue, and therefore, when we apply the color, it will restrict the staining to a particular section of the cell or the tissue, and it helps us to know what is happening. Now, on this slide, this is some cells were being stained, and we were interested in like a protein called GFAP. So they develop antibodies against the GFAP, and in the course of the staining process, the antibody bound to the antigen. Then we also, I said that the antibody coupled to the antibody is a color giving substance, a color expressing substance. So we expose these tissues to light, a particular light of wavelength, and it gives this kind of appearance. Now, this is, so we use a fluorescent light, but in this kind of staining, we use a normal white light. This is what we get. But it's also based on the same principle, antigen antibody reaction. Then also in the process of staining, in the process of staining, sometimes we can stain deeper. Then we try to reduce the staining intensity, or we can decide to stain it lighter and try to increase the staining intensity until we get to the desired staining. So when we stain it deeper and try to take off some of the stain, we call it retrogressive staining. But when we stain it lighter and we try to increase it with time, we call it the progressive staining. Now, having said that, I will take you through the steps that are involved in now staining that glass like. So, it is made up of wax. And the first thing is that the staining is soluble in water. And the first thing is to take out the wax. And we usually use xylene in the lab to take out the wax. The next step is that after taking out the wax, the tissue, what we have is dry. And these dyes are water soluble, they are water-based dyes. So what we do is that we rehydrate. When we want to rehydrate the tissue, we pass the slice in decreasing gradient concentrations of ethanol. So when we were dehydrating, we started from lower concentration to higher concentration. But when we are rehydrating, we pass it from higher concentration to lower concentration. Then we wash the tissues in water. We see, because there will be alcohol and we need to get rid of the alcohol. So we rather we pass it through water and rinse the tissue of the ethanol. Then we now do the first staining. Usually we stain the nucleus first. Hematoxylin is a very deep dye, very, very strong dye. So we do that. We have a particular time to do it. Then we can wash off the after some time, we wash off the excess stain in water. We wash off the excess stain in water. Then we do retrogressive staining by the process of differentiation. Differentiation takes out some amount of the hematoxylin so that the hematoxylin will not be too deep. Then we also do some form of washing and take out. Please, can you hear me?
Yes, sir.
Yes,
Yes,
sir.
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 Yes, sir.
Yes, please.
Okay, okay. So we wash off the water, and the washing is also gentle. We don't do it rigorous. We do a gentle washing of water in water. Then before we bring in the cytoplasm stain, so we stain the cytoplasm, which is the second stain. Also for some minutes, then we also wash off the excess stain in water. Then we now do a dehydration. We dehydrate it because we stain it. So now we don't need the water to be present. We take out, we pass it through increasing gradient concentrations of ethanol. Then also the next step is to get rid of the ethanol in a clearing process. So we use xylene for dewaxing and we use xylene for clearing. The last step is to mount. We need to mount the slides, the stain slides. And it is a final stage. And in the mounting, what we use is that we use an adhesive medium. We use an adhesive medium to fix a thin glass slide over the stained tissue. And this is a adhesive medium, the mountant. This mountant has a refractive index which is nearly equal to that of ordinary glass, similar to that of ordinary glass. So, and it allows light to pass through. There are a number of mountants. Some are water-based and others are not water-based. So then those which are not water-based, we have the DPX, we have Canada Balsam, we have Acrytol Sub X. But those which are water-based, we have the clear mount. Okay, depending on if we want to keep the slides for a long time, it's better to use a non-aqueous base. And if you want to use a non-aqueous base, the same section should be cleared of water. Okay, so in mounting, what we do is that we apply a single drop of mounting on the medium. Then we hold the cover slip about 45 degrees, allow the drop to spread along the edge of the slip. Then we gradually lower it and allow the mountain to spread slowly. Then we also we are mindful of water. Sometimes the bubbles, because maybe we may not have removed. When air can also pass through the mountains and create bubbles, so we get rid of the bubbles. Other than that, someone will see when the bubbles are there, they block the view of light, and you will not be able to see the tissue. So we also get rid of the bubbles. Then we can have a very clean slide. We allow it to dry, then it is ready for use. So the slides, what we are described is actually a permanent slide, but we also have a situation where we can prepare temporary ones. So these are usually needed for short periods, usually used within a day and discarded after examination. And usually you can mount it in water or another fluid of low volatility. And one thing about the mountant, the temporary permanent slide, in the lab we have mountant permanent slides which dates back to I think 2011. So that's 14 years. We have those slides still in the lab. So if we want to prepare them for longer time, we use a permanent preparation. Okay, so that is what I described. On the slide, I have added a video which is going to summarize whatever thing that we've done. So just listen to it and let's see. After that, we have some questions. Please, can you? Yeah.
He said we cannot hear.
So please, the volume is not working also.
The video is showing, but there's no sound.
I can hear you.
Please, we can't hear.
Yeah, hello.
Hi, sir.
Okay. Okay.
Hi.
I will send the video to you. So no problem. I'll send the video to you. Okay. So maybe if you are struggling, maybe you can pause. We can pause and let's reconvene. So let's can take a break. 9:30. We can continue with the last section. Okay. I'll get the video sent to you so that you can play it and listen to it. Then 9:30 we continue. I hope that this arrangement helps.
Yes, sir.
Yes,
Okay,
Doc.
Yes, sir.
Yes.
Yes, sir.
Okay, so let's reconvene at 9:30.
Please, is the class over?
Please, is he back?
No, please.
Okay. Thank you very much.
Yeah, hello. Yeah,
Hello.
yes, I think I have a problem with my computer, but I'm sorting it out. Okay, so I'm sure.
Oh, okay.
Yeah,
Okay.
so just exercise patience.
Okay. All right.
Ebusua mpacho lecture na waba na.
Or say when your problem on the computer or ba.
Oh,
The man
okay,
time
okay,
kakra.
yo, me das.
Sorry, please, can you see my screen?
Yes. Yes.
Okay,
Please.
thank you. So this last section will give an overview of like light microscopy, and I would mention that at the end of the lecture, maybe you should be able to mention the functions of the parts of the light microscope, and should be able to recall how to use a light microscope. Okay, so maybe you can also mute so that the microscope has become one of the most recognized. It's like it's a symbol of science, and especially if you are doing detailed science, you cannot do it without the microscope. And the microscope started as a simple lens. So I'm sure we've seen the magnifying glass. Then later we got a lens which can do it detailed. So it started with the use of simple lens in the ancient times and we now got what we call the compound microscope. It's around 1590. And it is interesting to know that we've made a lot of discoveries about what you call the invisible world. That is the part that goes beyond our eyes. The microscope, what it does is that it magnifies or enlarges the image of very tiny objects. And even now, we can have currently the basic microscope in the lab at least can magnify objects 40, 100, or 400 times. And even this microscope, we couple them with cameras, and these cameras can help us to magnify the object even like almost 10,000 times. There are different types of microscope, and one of the criteria is the source of energy that is used to view the object. So we have, like for example, light microscope, which with a light microscope, it uses light as a source of energy. So usually you have a bulb, a white bulb, which will be the source of energy. Then we also have a situation where electrons, like what we see in the cathode ray. If you remember the cathode ray, it generates electrons. An electron beam will hit in the object, and that will also be a form of energy. When it hits the object, then it will generate images out of it. And therefore, that also gives us what you call the electron microscopy. So I'll comment on the light microscope, which will be the main focus, the focus of this lecture. So light microscopes are compound microscopes. And they are compound microscopes because they use more than one lens. So they use more than one lens, and that's why they are compound microscopes. If they use just one lens, that would be a simple microscope. So in the lab, the light microscopes that we have, they are all using even the simplest of them is a compound microscope. So they contain two lenses for magnifying an object. So we have one of the lens which is very close to the specimen. That is the objective lens. Then we have the other one, that is the eyepiece. That is the ocular lens. And even we can further classify this compound microscope into two based on the number of eyepiece lens. So if it has just one eyepiece lens, we say we are dealing with monocular microscope. But if you are dealing with two eyepieces, then we say it's a binocular. So we have a binocular light microscope and monocular light microscope. And even there are also other criteria. We have like we even have what you call the upright and inverted. Now these microscopes are upright. You see, they are upright in the sense that you have the lenses just on top of the stage where you put your image. You have it just on top of the stage where you put your image. We have a situation where the images are rather um, above the object. I think I want to see if I can just get a quick image for you, so that for clarity's sake. Let me see if I can get an image for you. Yes, please exercise some patience. Let me get. So we have also others where the image, the sample is rather on top. So that is what gives us the inverted microscope. Just a moment. Please, can you see the microscope?
Yes, sir.
Oh, please.
Yes, sir.
Okay. So with this microscope, okay, maybe I can put the other one side by side first. Okay. So you see, with this microscope, I think can we get the I think the person can mute himself.
Nuruddin, mute yourself. Nuruddin, mute yourself.
So if you look at this microscope, you have the stage, and these are the objective lenses on top. But if you come to this microscope, the objective lenses are here and the stage is above. So that is a so it's a kind of different. Then we have also dark field, bright field. Sometimes you look at the images and you, like in this case, the image is having a white background. So, but we also have other instances where the background will be black, and that would be a dark field. So these are the various types of microscopes. We can talk about light microscopes. Please, can you confirm whether you can hear me? One person.
So we can hear you.
Yes,
Yes.
please. We can hear you.
Okay, thank you. So we have the light microscope, and we look at the various parts. So this is the eyepiece, which has and there's a body tube within which contains a lens. Then we have the arm where you can hold. We have a base. Then we can talk about the lenses. So we have the objective lens, that is the eyepiece lens, and the objective lens are stuck into the nose piece. And because you can swing the nose piece, we have it's a revolving, so you can turn it to determine which kind of objective lens that you use. Usually the objective is changeable, but the eyepiece is fixed. Then we have a stage. Stage, we have a stage clip. This for some microscopes, the stage is print and the stage is adjustable. You can move it in both up and down, vertical axis, and also in the horizontal axis. But with this kind of microscope, you cannot adjust the stage in the horizontal and the y-axis, but you can move it. We use a coarse focus and the fine focus. Sometimes we use the word. The popular one is the coarse adjustment knob and the fine adjustment knob. We use it to move the stage up and down. Underneath, we have also the diaphragm, which will regulate amount of light in it, and this is the light source. Here on the base, we can have a switch to the light. We can have a switch to the light. Sometimes there can also be a knob. There is a knob that will regulate how much light, the light intensity. So this is a part of the light microscope. So we take the parts one by one and learn them. So we have the eyepiece lens, which is also known as the ocular lens, the body tube. So this is the first lens that one looks through. It's about 10 times. There are lenses, and therefore when light passes through, the light will be refracted, so that the object which is underneath them will appear closer. Then we have the distance. We also have for the eyepiece the binocular, and sometimes you can adjust the distance between the binocular lenses, so that when you are looking through it, it will appear as one field of view. In that case, when you are looking at it, your eyes are not strained. Okay, so when you adjust, you can move them sideways to adjust the distance between the two eyes, and the field of view will appear as just a single circle, and one can also view the field with less eye strain. Then we have the objective lens. The objective lens is used in combination with the eyepiece lens, so that you can provide a range of magnification, and it is located below the nose piece. So it is inserted into the nose piece, and you use the nose piece to determine which kind of objective lens that you want to use. Usually, when you hear a click sound, then you notice that the objective lens is in phase. If you don't hear the click sound, the objective lens will not be in line with the eyepiece lens. So when you observe you look through the eyepiece lens, you will not see anything. Okay. So now the objective lens, they have different magnifications. You have one with the red ring. This is a universal color code. So it doesn't matter where you go, you find it. So you have a red ring, which can magnify the object by 4 times. Then we have a yellow ring, which is 10 times. The blue, which is 40 times. Then there is another one at the back there, the gray ring, which is about 100 times. And usually we use the 100 times together with oil so that it can help in the image. Then we have the nose piece, which holds the objective lenses. You rotate to enable the use of a particular objective lens. Can talk the base. The base is a major support of the whole of the microscope. And if you are carrying the microscope, you need to hold that place. You can place always have one hand on the arm and one hand on the base so that it doesn't fall off. The arm also support the upper part of the microscope, and just like the base, we use it in order to be able to hold the hand. Then we have the stage. So the stage is where we put our slide, and the slide contains the specimen. So that one is viewed with the microscope. Usually the stage can be, the stage is movable. Okay, but however, the clips, the range of movement is where it's a problem. So some stage, you can move the stage up and down and also sideways. So the stage is always movable so that you can be able to focus the object. But as to whether you can move it left, right, that is the x-axis, that is also another issue. So that is where the stage is different. Okay, so the movable ones are those that you can move in the XY plane, like this microscope. So this allows you to be able to move it. Usually there is a knob at which will protrude downward, which will enable us to see. Let me check if I have a picture like that. Like this one is, you can see this is the adjustable stage. It helps you to move it in the X and Y direction. So this is a movable stage. Okay, I will update the slide so that we can get yeah, so maybe control Z, control C. So I would we have this. So we can pin add animation this again. Yeah, so we have the stage. So and on the stage we have the stage clip, which will hold the stage, and there is an opening in the stage where light will pass through. So this is the one with the movable stage. So like this is a stage adjustment knob, which usually there are 2 of them. It allows you to move it in this direction, the X Y direction, and another can let you move the stage in this direction. So with this, you can actually view various parts of your slide. Okay, so the movable stage has a stage control knob that allows you to slide in the X and the Y. Other than that, if it is fixed, where you position the slide is where you can view at that time. And many of the labs are having this kind of adjustable, the movable stage. Then I've indicated that a stage clip which is used to hold the slide in place. Usually one on each end. They are different design. Like in this design, you lift them, then you place the clip on the slide. But for this one, like this one, you place the slide here, then on one end, then you use the other end, which is a movable clip. Then you use it to clip the further position, the anchor the slide. So they are different designs. Yeah, so that is the stage control knob, which I indicated. So that's a stage control knob. With this, and we can also move this one. Then we have the light source, which will provide light for viewing the object, the specimen. Usually, the old microscopes, there was a mirror. They use a mirror, but this one is an illuminator. But it sends light through the opening. And usually the microscope has a switch for the light source, like here. So when you switch it off, the light will go off. When you switch it on, the light will come on. And there is also a control to regulate the light intensity, how much light should be passing through it and otherwise. Then there is also a diaphragm. It's below the stage, and it also is a wheel or lever which is located below the stage opening. It also regulates the amount of light that can enter the lenses. So we do that hand in hand. Then we have the coarse adjustment knob, a very big knob, and a fine adjustment knob. They are separate, but in some microscopes, the coarse adjustment knob and the fine adjustment knob have been put together. And the coarse adjustment knob is the bigger side of the knob, while the fine adjustment knob is the smaller side. So we use the coarse adjustment knob to raise the stage through a relatively large distance, whereas the fine adjustment knob will use it for small distance. Usually the fine adjustment knob is usually very useful for focusing and helps you to get a very sharp view. And I've indicated that they vary in the arrangement of the coarse and fine adjustment knob. Like here, you find the coarse adjustment knob and the fine adjustment knob fused, and the coarse adjustment knob is a bigger one. Fine adjustment knob is a smaller one. So the last stage we'll talk about using the light microscope, where you are supposed to connect the power supply of the microscope and you turn on. Then you can press on the light switch of the microscope to turn on the light. You can use the controls of the light to adjust the intensity of the light. Now, when you are using the microscope, we advise that you always start with a ×4. So you use a nose piece to position the ×4 in place. Always make sure that the stage is positioned quite some distance from the objective lens. The stage that is usually further, the distance between the objective lens and the stage, the lens and the stage will be bigger, is the ×4. All others, the distance is very, very close for you to be able to see through. The distance between the objective lens and the stage should be very, very small. But with the ×4, the distance is relatively wide. I have been into a lab where the person was using ×40, the blue ring. And the distance between the blue ring objective and the stage was very, very wide. I automatically knew that this person is not seeing anything. Okay. You can use the coarse adjustment knob to lower the stage. You rotate the nose piece such that the ×4 objective lens is directly above the center hole of the stage, and a click sound will let you always know that the objective lens is in the right position. Then you place the glass slide in the center of the field of view and you secure it with the stage clips. You should bear in mind that the cover slip should always face up, and you also have to adjust it so that the stain segment, the light will pass through the stain segment section of the slide. Then this will allow you let the light pass through the tissue to be viewed. You look through the eyepiece to observe the illuminated section. Then you rotate the coarse adjustment up. When you rotate it towards your head, the stage will move up. And when you move, rotate the stage adjustment knob away from your head, the stage will move downwards. But you need to let the stage come closer to you so that you can put the image in focus. Then you can use the fine in adjustment knob when the view is clear and you can get a very sharp object. Now, once you are focused with the ×4, you rotate the nose piece and switch to ×10, and you can repeat the steps 5 to 7. Then you can now switch to a high power, which is a ×40. Usually, at this point, only use a fine adjustment knob. You don't need to use a coarse adjustment knob. So these are the steps in using the light microscope. I think we made a point that it's practical. So if you think through a possible practical that you can do. The biology, they have not decoupled the practical from the theory, so it makes it quite difficult. So I'll find out. I'll think through if we come up with a very good practical schedule, which can be done across all centers. I think, how many centers do you have? Wesco and I think UCC. And may I know, those doing online, which centers, where are you based?
We have time at Cambodia. Cambodia, we have time as well.
Tamale.
So I will ask. I will engage the office. Yeah. So I will engage the office to have an idea of where you are based, and if we have a card that we organize, we will see if it's feasible. We'll gladly organize that. Okay. Okay. So this will bring me to the end of today's class. Yeah. So I don't know if there are no questions. Then we can reschedule.
Yes, sir. Please, I would like to find out if this particular session of our lecture has been recorded, and the transcript matter will be sent to us.
I think
sorry.
I noticed that code was recording. So please contact Madam Rebecca to help in that sense.
So that if we should go to YouTube with a code, we'll be able to.
Yes, I think they have recorded it.
Sir, please, we would be glad if you can send the slide to our platform.
Yeah, the slide. One of you, I will send it to Rebecca. But if you, I think someone can also pick my number and send me a WhatsApp so that the class rep can pick it, so that I can send the PPT to him. So the number is.
Please, I have a question.
Yes.
Hello, sir.
Please, I'm listening.
Yes, sir. Please,
yes.
talking of the microscope, is there a way we can maintain or take care of the microscope, maintaining the microscope?
Yes, so there are several ways of maintaining the microscope. So, for example, the glasses, you see, it uses glasses, and it is not advisable to touch the glass surfaces with your hand. Okay, and also when you have a tissue wipe, a lens cleaner, which you can clean to, you can also clean the surfaces. If you have oil stains on the glass slides, you don't get a clear image, you get a blurred image. So that is also one of the things. Usually, another thing is that some people may, the stage, you always make sure that after using, you move the stage, use the course adjustment knob to move the stage downwards so that it is far. And you put, you swing back the ×40 in position. Always it's advisable, since that is far from the stage. There are some people, when they use it and they end up using the ×40, they'll just leave the microscope just in that position. and people who come to use it later may not be experienced, and they may end up breaking the the lens. Okay. Another thing is your electrical. You have to make sure there is a voltage that we use. So make sure that the voltage is we are using the right voltage. Other than that, the electrical circuit can get blown. Now, when you are holding the microscope, you have to use it in two hands so that at least you cannot. It may fall. We have a head. This kind of head. This head can be moved, the binocular head, and also you have to make sure that all the that head is in fixed position, including all screws, so that they are. Finally, when you are keeping the microscope, you keep it in a dry place, because when it is moist, mold can grow on the glass. And we even have a cover for the microscopes. You can cover them so that at least the glasses will not be tainted. So there are a number of practices that we do to care for the microscope. Yeah, I don't know whether I have a list there, but if I have a list, maybe I should post it, update the slides, and send you.
Okay, sir. Thank you.
Okay. Okay. So in the absence of further questions, I think we are closed for the day. So God willing, next week we will just like I send we'll start looking at the basic animal tissues. So we start with the epithelium, then later we continue the connective tissue. Then the following week, God willing, we do the nervous tissue and the muscle tissue. Then we do the plant tissue. Then we end. Okay. Thank you very much for your attention.
Thank you, sir.
The person can contact me. I'll send the files to them.
Sir, please, are you our course coordinator?
Yes,
Okay.
I am a course coordinator.
Okay.
Ernest Amponse Siama is my name. I should have introduced myself. Sorry.
Sir, please, the number again.
It's on the slide 0246.
Okay.
0246 46 2434. Yeah, we all the centers are using the same slide. We are using the same slide for lectures. So you can also
If there are some past questions with regards to this particular
okay. Yes.
Please, I want to suggest this. This course, some of us are offering chemistry, some to biology. But the biology ones, we are doing cell and cytology and cell and tissue. But we have realized that from our timetable, Saturdays we have this cytology, and after that we don't have lectures again. And it's my candid opinion though. Is there any way we can have another session on, let's say, Saturday after 11:30 upwards? We, the biology students, we don't have any lectures.
Well, this is the timetable that they gave me, the schedule. But if it is Saturday, you can just notify me. The class, so you can. So that's where the rep comes in. So with the first meeting, I stick to the time that they give. But if you notice that there's a time on Saturday that is feasible, you can communicate to me, and I also look at my schedule.
Okay. Actually, the first block, I was the rep for only online centers. That's the reason why I'm asking. Only online centers.
So your name?
Festus.
Okay. Yeah, so I don't have a problem. Last year, last year, Dr. Abuaji was, I think Dr. Abuaji engaged you.
Yeah, last block, yeah.
Okay, so what will happen is that if there is an alternative date, just let me know, and I'll compare it with my schedule, and I'll confirm. We usually don't encourage like 4-hour lecture continuous because you may also be tired. So if there is a time in Saturday, just let me know so that on the Sunday we can also we can split into two.
Okay, sir, please. So, now that we are on here, I don't know if you will permit me so that we have that discussion now. Now that we are done with your section. They are all here. I don't know if you can permit me.
Oh, you
have
can
that.
discuss it, but I'm sure you have a platform that you can. But okay, so you can start with the discussion. Yeah.
Okay.
Yeah, please. We also have chemistry after the cytology, so we can't be available.
Those doing chemistry, do you do this same course? That's a
Yes.
Yes, they do the
Yes,
same.
okay. Oh, okay. I never
Yes,
knew.
you all do the same.
So the splitting
So,
on Saturday will be a little bit difficult because of them.
so the only difference for this blog is the cytology, right?
No, soil.
The soil.
Okay. Oh, okay. I've understood.
But which people are doing the soil? The soil is for the biology major, Abi.
Yeah.
Yes.
Biology.
Major
biology.
biology.
Okay. Understood. Then it means that we have to maintain this time.
It's better we just maintain it this way because of them.
I say more. Okay. And
Doesn't mean that biology for the air focus. Yeah, right.
Yeah, four
Yeah.
courses
Biology
for this block.
major is 4 courses for this block.
Okay, all right, thank you.
And chemistry is 5.
I think so.
15 chemistry environment.
Chemistry for you. No, I'm gonna biology.
Cell and tissue organization. Data collection and analysis. Cytology, basic genetics, main group chemistry.
Oh, okay. I see. Thank you.